Welcome back to my channel, and as this is my first video, I am going to do the lesson Microorganisms, grade 9 students first lesson, first chapter. And today, as I told you, as this is a long lesson, I have divided it into two groups. In this video, I am going to show you up to the applications of microorganisms in agriculture, and in the second video I will show you continue from applications of microorganisms in medical field. So now today I'm going to do this presentation up to agriculture's application of microorganisms in agriculture. So before going to my video, I would like to request from you all, don't forget to like, comment, share and subscribe, and let's go to this.
Microorganisms. Who are microorganisms? Unicellular multicellular organisms which are invisible for the naked eye when taken individually is known as microorganisms. A microorganism or a microbe is a microorganism which may exist in its single-celled form or a colony of cells. As a single cell is similar to unicellular, and colony of cell is similar to multicellular. When we are doing science, unicellular and multicellular are the scientific words that we use when doing science. So let's go. Who observed the microorganisms first? This is a main factor that you should take as general knowledge. The first person who reviewed that there are microbes living in this world, and the first person who reviewed that there should be equipment to observe these microorganisms because some are visible to naked eye but some are not. By considering all these factors, the Dutch scientist Sir Antonio van Leeuwenhoek observed the microorganisms for the very first time using a microscope, using a special microscope called light microscope. So this is the microscope that Antonio van Leeuwenhoek used to observe the microorganisms for the very first time. So you can see here a picture of Sir Antonio van Leeuwenhoek. He is not a just scientist, but he is a Dutch scientist. That is also a main fact that you should remember. So two types of microorganisms. As I told, there are two types: unicellular microbians and multicellular microbians. Unicellular microbians means single-celled microbians, as I told you in the previous slide, and multicellular microbians is the colony cell microorganism. So when talking about unicellular microbians, the definition for that is unicellular microorganisms are the microorganisms made up with only one cell, and multicellular microbians are the microorganisms made up with more than one cell. And the examples for unicellular unicellular microbes are protozoas and some of the bacteria, and some of the bacteria consist to multicellular microbes as well. So the examples for multicellular bacteria are protozoa, virus, and algae. So let's move on. You can see a picture of the organisms classified as unicellular organisms and multicellular organisms. So there are mainly 5 types of microbes. First one is bacteria, second one virus, third one protozoa, fourth one fungi, fifth one algae. You can see here this is a picture of bacteria and this is virus, this is protozoa, this is fungi, we normally call it as mushroom, and this is algae. So when talking about the features of bacteria, they are unicellular and microscopic. As I told you, some of the bacteria are unicellular, but some are multicellular. Bacteria are made in 4 shapes. Spiral shape. The first one is spiral shape, and second one is comma shape. Third one is spherical shape, and fourth one is rod shape. So in front of these shapes, you can see another names. These are the examples for these shapes microorganisms. The examples for spiral shaped microorganism is spirilla. For comma shaped, the microorganisms is vibrias. For the example for spherical shaped microbe is quasi, and the example for rod shaped microorganism is bacil. And there's one more feature of bacteria. They widely spreads in every environment. So you can see some pictures of bacteria. Examples for bacteria are Lactobacillus bulgaricus, Bacillus anthrax, and Vibrio cholera. Not only that, we have Streptococcus thermophilus as well. So the second type is virus. They are electromicroscopic. We cannot observe through virus through light microscope. We should use electromicroscope to observe virus. So second one is multiply only within living organisms. This means they reproduce only in living organisms. Third one, display both living and non-living characteristics. The one living feature that virus shows is they do reproduction, but the non-living characteristic is they do not show locomotion. That's a difference between virus. So virus cannot be considered as a uh, living organism or non-living organism. And the other one is no cellular organization. And the fifth one is they do not show other living characteristics like respiration and locomotion. Either than these two, they show other living characteristics. Examples for virus are HIV virus, influenza virus, Ebola virus. And today the pending virus, corona, is also considered as a virus. So beneficial effects of virus are, there are mainly two beneficial effects of virus. First one is genetic engineering. Genetic engineering means virus can be used to transfer germs from one organism to another organism in genetic engineering. The main example for genetic engineering is human insulin is transferred into E. coli virus. And the second factor is control some infections. Viruses are used to control some bacterial infections. So you can see after a genetic modification of apple, it has turned into insect-resistant fruit. That's the difference of genetic modification. And when talking about protozoa, they are also unicellular and microscopic. You use structures like cilia, pseudopodia, and flagella for their locomotion. Live in aquatic environment as well as in other organisms. So this is a protozoa. The protozoa mostly live in aquatic environments, but not only in aquatic environments like as virus. They live in organisms as well. So the other one is fungi. Fungi can be seen in unicellular as well as multicellular. Some reproductive organs of microorganisms can be seen with naked eye. You can see here some, we simply call this as mushrooms. This is the reproductive organ of this fungi. And they substrate on moist surfaces. Substrate means they thrive on moist surfaces. And the other one is algae. Algae is unicellular and multicellular. As we know, algae is green in color, so the plants with green in color we known as chlorophyll plants. So they possess chlorophyll and they have the ability to do the process of photosynthesis. And you can see some algae varieties like ulva can be seen with naked eye without any microscopes. And not only that, some algae varieties are floating on water and they are simply known as planktons. And that is also a major point that you should keep in mind. And the other one is beneficial effects of microorganisms. As everything in the world, microorganisms also have a beneficial side and adverse side. So when talking about beneficial side, microorganisms are used mainly in agriculture, medicine, and conservation of environment. Not only that, microorganisms are used in industries in sake of economical advantages and for research purposes. This is also the places where microorganisms are used for beneficially. So you can see here for yogurt production, microorganisms are used. When talking about agricultural field where microorganisms are used, the first factor is gene technology. In agriculture, crop harvest is enhanced and enriched by producing drought-resistant and pest-resistant crops with high nutrients and taste, with the aid of microorganisms. Further, biological pesticides and weedicides are developed using microorganisms. As a major example, the gene technology is used to convert normal rice into golden rice. As I told you, the major place where gene technology is used in by using bacteria is human insulin is transferred into E. coli virus. And even in agriculture, normal rice is converted into golden rice, which is enriched with vitamin A by using gene technology. Golden rice is enriched with vitamin A, and it has uh, it is very nutritious for human body. And you can see here photo about more information about gene technology. And the second factor where microbes are used in agriculture is nitrogen fixation. As we know, there are 78% of the nitrogen in the atmosphere. As well as we learned in grade 7 that there is a generation known as leguminous in plants. So the plants which contains to the leguminous generation have knots in their roots, and they are we simply known as root nodules. That we learned it in grade 7. So in these root nodules there is a bacteria known as rhizobium, and this rhizobium helps this is a nitrogen fixing bacteria, and this helps to absorb nitrogen from our atmosphere and make the plant fertilized. And this process is known as nitrogen fixation. This rhizobium is known as a bacteria, and the main bacteria used for nitrogen fixation is rhizobium. Let's move on. And here you can see there's root nodules, and inside these root nodules there's microorganisms bacterium known as rhizobium, and the rhizobium helps the root nodules to absorb nitrogen from the air. And third one is what are biofertilizers? Biofertilizers is microorganisms are used as biofertilizers in agricultural feed as a third point. So biofertilizers means, as we know, Acidobacter is also a free-living bacteria like Rhizobium. Acidobacter is also a nitrogen-fixing bacteria like as Rhizobium. So in today, with the development of the technology, Acidobacter bacteria is cultivated in farms to make their plants fertilized, and this is called biofertilizers. Acidobacter who lives freely in the environment, and nitrogen fixing bacteria which is used as a fertilizer for the plant is known as biofertilizers. And the other one is producing compost. Compost is a decomposition of organic matters. To the decomposition of organic matter, when we use microbes for the decomposition of these organic matters, this process becomes rapid. This process happens rapidly. So when we use microorganisms for this process without using any chemicals, it does not damage the environment or the atmosphere. So without damaging them, without damaging It adds minerals and vitamins to the soil in these organic matters. So that's the reason where when there are when plants grow on compass layers, because under the compass layer there's many vitamins and minerals underneath of this compass layer. So mainly for the compass production, fungi and bacteria are mainly used. So you can see here a picture of decomposition of organic matters and biopesticide. The fourth point that why microbials are used in agricultural field is biopesticides. We know who are pests. Pests are the harmful animals that damages the cultivated crops. So to control these pests, we use pesticides. Pesticides means the things that we use to destroy pests. So the biological pesticide is simply known as biopesticide. It means that the destroying of pests in biological method. This is the meaning of biopesticides. So the biopesticides, the definition is some of the microorganisms can be used as biopesticides to control insect pests that cause damages to the crops. The main example in agriculture is the fungus Alternaria is used to combat the aquatic weed Salvinia.
Now I think it is crystal clear for you about applications of microorganisms in agricultural field. Up to that, I think now you are well cleared about that. So now I'm going to end up my video. So I will bring up you the microorganisms part 2 quickly. So until next time, goodbye.